clarity significantly. We had about a 5 foot increase in water clarity depth compared to the 30-year median. So we had a median of 24.2 feet versus the 30-year median of 19.6 feet. So as we remove the cyanobacteria, the water clarity improves because the cyanobacteria are suspended solids. They kind of make the water cloudy. We had based upon measurements that I did, and I hope if people have questions about that, I can elaborate on it. But we did some intensive spatial and temporal assessments of White Pond's water quality and cyanohabs, some cyanobacteria. I call them cyanohabs, an acronym they use. Stands for cyanobacteria, kind of blends with harmful algae blooms, become cyanohabs. So you'll hear that again from other people. But we had a 51% reduction. So as the APOD was doing its work, removing the cyanobacteria, we saw a corresponding decrease in the overall biomass and actual bio volume, the biomass over the water body volume of White Pond of 51%, which is pretty significant. And that also corresponded to a decrease in nutrient concentration of the cyanobacteria itself. So as we remove the cyanobacteria, the populations that remained struggled to get whatever nutrients they could, and we saw a decrease in what they could get as time went by, which is one of the metrics we used as part of our analysis. And so over that 12-month period, we removed just under 400 pounds of moist to dry cyanohabs, and we composted them, we biodegraded them in a controlled manner, which also reduces the cyanotoxins significantly. Let's see if I have any other points here. But that's primarily the big talking points, what everything means in terms of all the nitty-gritty data that I collected. That we did see an increase in water clarity, reduction of nutrient concentration of cyanohabs themselves and in the sediment. And we saw an overall decrease of about 50% of the cyanobacteria in the pond as a whole. So that's kind of the big picture perspective. I have a lot of other data we can talk to, but I guess I'd rather see what questions people might have.
John, were you going to show anything or share? You talked about a PowerPoint or the report itself?
Yeah, so I have a PowerPoint presentation I did on the 17th of the 16th of March. February. And I actually just I recorded it with my, with an oral presentation so people could understand it. I just finished that yesterday, but I'll post that on it's my sister technology website called ppodtech.com. So it's PPODTECH.com. I'll post that there for people to look at. It's a really large file. It's about 80 megabytes because of the photos, but I can post it on my website and people can look at it there.
Can you say that? Yeah, he's going to post the video. John, could you give us the website address again, please, for our minutes?
Yeah, it's www.ppodtech.com. So it's ppodtech.com.
Great, thank you. So I guess we'll start with the people in the room here. Does anybody have any questions for John Case in the report? Could you state your name and address? That's what we do for everybody who's on the team.
Alyssa Brown, private company. Can you explain the role of sulfur in connection?
Yeah. So sulfur is a nutrient. It's a macronutrient for cyanohabs themselves, as well as other plants. It's a common ingredient of fertilizers for farmers and things like that. But it's also a biogeochemically active nutrient, in that its presence affects the ability of other nutrients like iron and phosphorus to make them more available than they otherwise would be. And so what we're living in a consequence of the acid rain deposition, which is actually nitrates and sulfates, And it's why we passed the Clean Air Act Amendment of 1990 was to reduce the amount of sulfur emissions in coal. But that's had a significant change in our environment that actually kind of predates most of our knowledge. So it's kind of like we're used to a normal now that didn't really exist until the Industrial Revolution came along and all these sulfur entered our water bodies and forests and things like that. And sulfur is used in many different products today, mostly in detergents and fertilizers, and it just keeps the phosphorus that's out there more active than it otherwise would be and available for things like cyanohabs. So it's really the prize. The phosphorus itself, this is me talking about the data again, but so I did a sediment core sample. If people looked at the report, we had about a 2-foot recovery of soft sediment, which was chronologically dated by Steger et al. In a paper in 2020 on the cultural eutrophication of White Pond. It's linked in my report. But we were able to look at that and correspond the concentration of nutrients, including sulfur at depth. And it is concentrated right at the surface, and it's just not going to allow that phosphorus to otherwise be naturally deposited like it had been before the Industrial Revolution at White Pond. So phosphorus is, phosphorus and sulfur all have natural sources in ways of getting into White Pond. Some are unnatural, some are related to people. What we try to do is manage the things that we can manage, but you have to recognize that there's also these natural sources of nutrients, phosphorus in particular, that just will find their way into White Pond. But when you have an agent like sulfur, it's just going to keep that phosphorus more available than it otherwise would be. And it also, sulfur also increases the biodegradation rate of organic material. So as it falls down, the detrital material, it gets biodegraded that much faster, even in the absence of oxygen. And I don't want to talk forever about sulfur, but sulfur is also an oxygen scavenger. So a sulfide will scavenge any available oxygen, turn itself into sulfate. So it also creates anoxic conditions. And the list goes on, but that's the nutshell there.
So John, you mentioned in the past that you had theorized that sulfur could have been one of the big contributing problems to the cyanobacteria. Is there any methods that when you're collecting cyanobacteria, are you actually removing any sulfur at the same time?
Yeah, we removed quite a bit of sulfur. It is a macronutrient, so by weight, it's about equivalent in terms of need for a cyanobacteria as phosphorus is. So it's like almost a 1-to-1 requirement of the cyanobacteria to have as much phosphorus as it does sulfur. In a nutrient, by definition, what a nutrient is, and sulfur is a nutrient, it assists with the metabolic function of an organism. It's essential in order for it to complete some activity like photosynthesis. So, but we had a reduction in sulfur content with the cyanobacteria. I don't have the slide in front of me, but so that is one of the benefits of actually removing the cyanobacteria. Is you actually remove all the nutrients that it always needs to survive, or at least thrive at nuisance concentrations.
So then my next question is, I know that there's a plan for a couple of APODs in the water next summer, or this coming fall, whatever, spring. But would you ever, I know you have a subject with SPOD as well, would something like that be beneficial for a sediment?
Well, I put that out there as part of my recommendations for the town of Concord, you folks to think about. So there is a way of removing those excess nutrients that may go beyond what the cyanobacteria themselves can take in. And that those are either my SPOD or the PPOD. They're different technologies. There's actually summaries of those on the ppodtech.com website. But so the SPOD, S stands for sediment, and that would be used to help remove the upper layer of really soft sediment. And unlike other dredging techniques, which is basically what it is, you can lower from a raft down to the sediment surface, say 64 feet down, and remove just that sediment without, dewatering or having huge land-based infrastructure. So it is a way of kind of forensically targeting and removing those soft sediments. And the pea pod actually creates these geochemical conditions. I know it sounds all nerdy now, but it allows me to have an enclosure where I can control the geochemistry in an area to make the in this case the sulfur soluble, so that it would be contained in the water within that pea pod, and I could extract it that way without doing any kind of sediment removal. So those are just things to think about, because it's your kind of it's the sulfur it's an agent that just lies there and creates difficulties no matter what else you do. And so with phosphorus in White Pond, and this actually includes Walden Pond, a big source of the phosphorus is actually pollen, or what you call dry deposition dust, that just happens to settle in there. I think last year, if people remember those pollen storms that you had, we had a lot of phosphorus finding its way into White Pond that way. And so when you have those natural sources entering the pond, regardless of what anybody else could possibly do, with an agent that keeps it more available than otherwise would be, that's when you have issues like with cyanobacteria, or you could. So
Thank you.
you're welcome.
Yeah, this is Don Coleman from 55 Central Street. I had thought that there was less sulfur deposition these days because it was scrubbed on the both power plant.
I'm sorry. Okay, go ahead. Sorry. Could you say that again?
I heard that there's less sulfur deposition, because acid rain is essentially going to roll back. And so I'm so surprised that you say that there's more sulfur.
Okay, so I didn't actually hear all of that, but I think I understand. So we had, and actually this goes back, if you read my report, and actually the PowerPoint slides that I did will talk a little bit more to some of the historical records for the Industrial Revolution impacts to White Pond. And actually goes back to Henry David Thoreau, actually, some of the observations he had in the mid 1800s of the water clarity of White Pond being remarkable, but having a slight greenish tinge. And I wouldn't attribute that to cyanobacteria at that time. I would actually attribute it to the aerosol, the deposition of the dust and things like that from the Industrial Revolution. So I didn't get your first name, but so what happens is once that sulfur gets into the system, it does tend to stay there. It gets recycled near the surface. And there's other geochemically active things like arsenic that have a similar pattern. And they'll just continue to get recycled at the surface layer. So, and there's other sources of sulfur too. I mean, there's auto emissions, and there's people, doing what people do, and lawn chemicals and fertilizers and things like that. Detergents are the biggie, actually.
It seems like a problem with cyanobacteria is the last 10, 15 years. In that period, what's changed that could cause the cyanobacteria to come up? And I would say it's not sulfur, and that sulfur has been going down.
No, so I don't want to keep pointing the finger at. So it is kind of a tipping point. And so we've had documentation, or you folks have had, not me, but the records that are reviewed for White Pond since the 1980s. There's been instances of algae blooms at times. And, it's become more frequently monitored in the past, say, 10 to 15 years. So I think we kind of hear more about it because people actually test for it more regularly than they had in the past. But, so as every year goes by, if you just say the source of, say, phosphorus is say, pollen from trees, each year you're going to get a little dose of extra pollen and phosphorus. But when you have the sulfur in the system, it's going to keep that phosphorus more available than otherwise would be. And we can actually see, we can see that in my report. If you look at the sediment summary table for its SED3 as the number, the lab sample. It's the profile we did. And it actually shows a fairly elevated concentration of phosphorus at depth, chronologically greater than 200 years ago, before the Industrial Revolution. And we saw an increase kind of during the beginning of the Industrial Revolution. And then this is talking phosphorus now. And then we actually saw a decrease toward the top of phosphorus. But we saw the opposite trend for sulfur, lower at depth, pre-Industrial Revolution, and then just increasing concentrations. So you actually doubled at the top 2-inch layer. So it's kind of like this, it just keeps stirring the pot. That's what sulfur tends to do. It keeps that phosphorus otherwise available, that otherwise it would naturally bury. And we have that data for White Pond to show that that's what it wants to do, but it's not able to.
Does anybody in the, thank you, Don, anybody in the Zoom virtual audience have a question? If so, raise your hand. And while I'm waiting, Don, I have another question. I know you had markers in the water, kind of watching for, wind or water flow direction. Can you talk a little bit about that and where it seems to go?
Yeah, and before I do, so I anticipated from the feedback I had while I was in the field in Concord that the level of interest and knowledge of the residents of Concord is pretty high compared to some other towns that I've worked in. And so I did include, for people that have other interests in looking at sulfur more in other things, I did include a pretty extensive reference and sources list in my report. And I included hyperlinks to those so people can find them and look at them at their own leisure and to their own interest level. So to get into Beth's question, so in White Pond, there are no inlets or outlets. And I mean, hydrologically, there's no streams flowing in or flowing out of the pond. And yet we have these fairly strong water currents in White Pond. And those are things that we measured with these little floating things that Beth mentioned that they're called drogues. And the term drogue actually means a sea anchor primarily. It's something that merchant marine vessels and, vessels away from shore would use to slow their drift. And what we do for hydrologic drogues that are used in White Pond is we are able to set this frictional mass at a given depth so that it creates a drag at that zone or that depth. And that'll track whatever currents are acting on it and pull around the buoy at the surface, small little buoy. And we track those over time, multiple events with GPS, and we also track them over distance to estimate the velocity. And at White Pond, we had the primary flow direction. And this is one of those things, like if you have a race around White Pond swimming, you want to know is it's clockwise. It's a clockwise direction. It's the primary water flow direction, and the water flow rate from the top 4 feet, 2 feet to 4 feet, it varies between 6 to 8 feet per minute with a greater velocity near the surface. In my PowerPoint presentation slides talk to that. It's not something that people really recognize. And when I mention it to some people that do mostly lake assessments, it's kind of like an unknown phenomenon to them. But from speaking to other geologists, you kind of get it pretty quickly. But it's actually a mathematical process that someone developed called ECVN. It's called the ECVN fluid flow. And it came about from when they actually mapped how icebergs flow around. It's a similar kind of concept. So when the wind blows, it creates friction, turbulent drag. And then as the wind fetch is sustained, it continues to create flow at deeper levels. And because water is incompressible, once you start to move it, it all starts to move. And then eventually you get this rotational pattern. And it can sustain itself for several days, even after the wind stops. And you'll see, if people have interest in that, they should look at the PowerPoint slides. I talk to that more, and I show the actual patterns. And we actually have patterns. So at the shallow flow from the center of the pond, actually flows outward radially to each edge. So there's another dynamic going on there too. So
Can I ask that, John, does that give you any clue or hint as to why we see more bloom, visible scum over by the White F side and by Thoreau Cove?
yeah, so it's a forcing mechanism. When that water flow pushes around, it's basically what it does. The cyanohabs, they're suspended solids, and they can only move up and down, and not in response to things like water current. It's just actually by photosynthesis, they gain and lose carbon and rise and fall in the water column. But as the water pushes them around, it'll force them together, which is when you'll see those concentrated green masses either under the water surface or on the water surface itself. And we actually see that in the apod, and I have pictures of that in my PowerPoint presentation. I couldn't include pictures in my report, the text. They would have made it too large for me to transmit to deal with you folks. So basically what the apod does is I can basically tell the cyanobacteria where it's going to be forced to, and that's the trap area of the apod. And I collect it that way. And all of our data that we collected showed a pretty significant concentration gradient of nutrients and cyanohab into the apod trap on most every day we're out there, actually. So.
Hi, John. Evan Ricker, 104 Bolton Street. Couple questions. So it's on the metric, the weight of the amount of cyanobacteria that was collected. Just help me scale that. What is that in terms of volume? What does that look like?
Well, you can actually look at it and say, what's the overall total volume available in White Pond? When I look at some of the lab data, we get it in micrograms per liter, and you can extrapolate that out. Not extrapolate, but you can calculate what that is in terms of the overall volume of White Pond. And I just have estimates of the overall water body volume of White Pond right now. But in terms of nutrient concentrations, or I should say cyanobacteria themselves, we're catching things beyond cyanobacteria, where, I would say White Pond, might have, for 2022, which is when I have data for 120 pounds of cyanohabs at any one time that's available. And they do grow each year. So, it doesn't, it's like a plant. You can harvest it, and if the roots or the seeds are in place, you'll get more the following year. And that's actually consistent with data from Walden Pond. So USGS did a really extensive study at Walden Pond, not for cyanobacteria. The test methods they had didn't really exist at that time, or at least their focal point. But they looked at overall phytoplankton, which includes good algae as well. And, they had about 220 pounds at Walden Pond, which is, essentially 3 times the size of White Pond in terms of volume and depth. So just to give you an idea. So in addition to cyanohabs at White Pond, we were definitely picking up pollen, and it's a good thing too, because it has a lot of phosphorus in it too, as well as some pine needles and little bits of just detrital stuff floating around. So if that helps kind of put things in perspective, but I do expect in 2023 that we'll get a new crop of cyanohab, a population basically, that'll go through the same seasonal changes that we saw in 2022. But what I do anticipate is that there'll be less of them. There'll be a less overall bio volume, which we already saw that from October 2021 compared to October 2022, which is apples to apples, same month, similar seasonal variants. I expect to see a decrease in overall bio volume in White Pond in 2023. I expect to see a decrease in nutrient concentration of the cyanohabs themselves, but I do expect them to grow out like any other plant basically does, say in July through the beginning of September. that's typically what they do. I think of them as like pumpkin plants. They kind of go crazy when you don't really look at them too long, and then they kind of diminish down. So
And so the location for the APOS, I think I heard you in the previous conversation. So is that determines, are you looking at the water flow direction? Are you looking at the prevailing wind? Are you looking at where the runoff is coming from? And you pick the most appropriate place.
yeah, so it is it's dependent upon number. Yes, not so much runoff. It's don't really focus on that too much because once it enters the pond, it actually rotates around pretty quickly anyway. So it's really about your overall position relative to the predominant wind fetch direction, as well as the water current. In addition to access, I mean, that's a place we can access that without really intruding on anybody else. It's a place we can park our car, walk down there. I'm not working in someone's backyard. I'm not working next to the swimming beach where, I might have people messing around with the APOD trap because you don't know what it is. So it's a pretty nice location right now in Thoreau's Cove. So.
And then, so it sounds like the first run of these met or exceeded expectations. Is that the case? Did it work? I guess sort of what I would look at is did the beef get forward or not? I'm sure you're looking at two different metrics on that. And two, if so, would then doing more of it, adding more, doing bigger, is there a way to sort of amplify the benefits?
So it worked really well in 2021, the fall in 2022. I was actually a bit concerned. So in August of 2021, we were seeing cyanohab scums over like 50% of White Pond. So, that to me looked like a pretty big task to tackle with the APODs, perfectly honest. We didn't have a lot of data at that point, other than historical data. We had no data on water currents. And so I didn't know what forces were going to work in our favor, basically. And then they favorably did work in our favor. But when we actually started in August of 2021. We had an APOD just off the south side of the beach, and it didn't work there. It just wasn't well positioned, and I did have people messing around with it a bit from just being near the beach area, but it just wasn't an advantageous area in terms of water current or fetch, wind fetch. So yeah, it was very favorable in 2022. I expect it to be similarly favorable because nothing really else changes here. We still have wind direction that goes pretty much to the northeast, and because of the bathymetry and the geomorphology of the pond, that creates those clockwise flows. And there's also another flow too, which affects more of the cyanobacteria, which causes it to push to the outside edge of the pond. And I can get really technical on that, but it actually is, from the best I can tell, it's related to an hypothesis that Einstein solved. Actually, it's called the tea leaf hypothesis. And that's where when you rotate the water and fluid with suspended solids in the tea leaves, in his particular instance, when you stop stirring, these frictional losses along the edge of the pond for us causes the suspended solids to move down, and then they move up and then when they get toward the center of the pond, they actually move up and kind of do that toroidal kind of flow. Anyway, so all that worked in our favor. It was really nice. I'm glad that it worked well for White Pond, and I expect it to do likewise this year.
2021 was a really dry, really wet year. 2022 was dry. So I'm wondering if part of the result would be this difference in that situation.
Okay, I heard the part about dry. So we had a reduction in water body volume in White Pond in 2022 compared to 2021. So I considered my estimate of the 51% reduction of cyanohab biomass or biovolume to be conservative. because we had less of them and less water body volume. So it's a conservative estimate. But I didn't really have all the question, but we also had some pretty significant rain events in 2022. And so it's every year is different. But as long as the sun's shining and the temperatures are fairly consistent, which they are, the cyanohabs, they've adapted for billions of years to take advantage and grow pretty prolifically when they have all the nutrients and the temperature and the sunlight that they need, which is certainly the case at White Pond. Did that help on the dry year?
I think the question, I'm kind of other voice saying it did. I think the question Don Coleman was asking was, he was stating that 21 was a much wetter year than 22. I don't know the average rainfall, but could this rain have an impact or not?
Well, so that's actually how we ended up at White Pond, because it was, and I like that analogy to the pumpkins, because in 2021 we had so much rain, and I know this from a fact because I know like Verrill Farms, they plowed over their pumpkin fields. They had so much water they had issues with, the soil microbial life just wasn't allowing the pumpkins to grow. And we had other locations as part of our National Science Foundation grant to evaluate, and we were just not getting the conditions that we were kind of hoping to find to do our research and development with the APOD on, except at White Pond. And there was a few others, but they just didn't match in terms of where we were in terms of schedule. But White Pond was one of the few ponds that did bloom out, so to speak, and allowed us to get some good data. But I'm not seeing, to his question, I don't see a change. We certainly didn't see a change in data in terms of performance of the APODs in 2021, which was a wet year, certainly, compared to 2022, which was a drier year than 2021. But we still had some significant rain events in 2022. But I didn't really see, it's a really deep pond. Having said that, White Pond is similar to Walden Pond, and about 40% of the overall water body volume in White Pond could very well be attributed to precipitation and retention over time, releases it slowly. So it is a significant component. That's why it fluctuates when you have wet or dry years as much as it does. But it doesn't, it's deep enough that cyanohab population just seems to be doing fine. It doesn't seem to care too much, whether water goes up or down.
Thank you. Any other questions before we invite Nancy to speak?
One more. So, did you do like a mass balance of like how much phosphorus is there in a given year, how much you took out in the APOD?
I only heard bits of that. So if you could repeat that, please.
How much phosphorus do you take out in the APOD compared to how much you take those in the pond?
All right, so our report talks about that in detail, and we measure the metrics that we use to evaluate how much we take out. Is we actually test the cyanohabs themselves, the solids, as we remove them. And then we also test the sediment concentrations in the pond, and our report includes that. And so, but the parts that we can't anticipate with a 1 year time frame, a short time frame, is like in 2022, we had a significant pollen population too. And so I think we had some phosphorus added in naturally to the pond that would have kind of changed how our data looked. But as it was, we had a 44% reduction in phosphorus concentration of the cyanohabs recovered over time. And we had about a 12% reduction in phosphorus concentration in the upper 2 inches of sediment. This is really deep sediment too. This is down, 62, 64 feet below the water surface. So that's still accessible to the cyanohabs. And I do expect to see, as the 2023 cyanohab population gets its legs, so to speak, and picks up whatever available nutrients it can, I expect to see a decrease very soon in the sediment concentration, just from that new population utilizing whatever it can get its hands on. But having said that, the phosphorus concentration in the top 2 inches is lower than what it is say 200 years ago in White Pond. So you do actually have these natural sources of phosphorus there. But the real question is, why is it so available? And I think that is the sulfur part of the equation. So I don't want to talk forever, but there are, if you look at my report, there are these naturally occurring elements, these iron ore elements that you'll get in anoxic and oxic waters that White Pond certainly had in the past that get affected by sulfur. And just sort of just a point of history for you folks, the Saugus Iron Works, people probably have heard of that. They actually moved to Concord when they ran out of iron ore around Saugus, and we're situated over by where Damon Mill is now. And so historically, we actually have these pretty significant iron ore minerals that would be natural sinks for phosphorus. They can hold up to about 4%, which is 40,000 milligrams per kilogram of phosphorus. And we don't even have those anymore. That I attribute to sulfur as well. But it goes back 200 years, so it's beyond our ability to understand that, unless you're like a geologist like me that looks into that stuff.
Thank you. One more question.
You mentioned the benthic metals in White Pond. And if you were going to do any manipulation of the sediment, big sediment, how would that interfere with that operation?
Okay, so I only heard part of it, but I definitely heard the benthic meadows and some sediment. And so what, again, let me ask you again so I make sure I have it. What was your question again?
Would sediment removal interfere with those metals?
Okay. Yeah, no, so sediment removal, if we did the S pod, it wouldn't, because it's a discrete area. It's a small device. It can be as big as you want, but it would be lowered at a depth where the Nitilla isn't present. So it wouldn't disturb them. And so it's an enclosed area, and it collapses. So you lower it down to the sediment surface. And again, on the peapodtech.com website, there's a little schematic of what that looks like. But you can lower it down from a raft that you can fix over the deep holes, the soft sediment, the anoxic areas where the Nitilla isn't present. Lower it down, disturb that sediment, and suck it out from inside this enclosed area. You'll see in the pictures from the website, and remove it that way. Or you could use the Peapod to actually just enclose that area of soft sediment. This is deeper than the Nitilla, which is that macroalgae, and you could extract those nutrients, including the sulfur, from those deeper sediments. It's all just geochemistry. But what I really like about both those technologies is, it's kind of my boss is to do no harm. That's why I always walk around. If I can come up with a technique that does no harm, that's what I try to do, and that's what both those do. So I wouldn't expect any harm to the Nitilla, and that's something I'd be very careful about. Any kind of activity at White Pond is try not to damage the Nitilla.
Great. Thank you, John. I don't know if you can hang on till after Nancy. Maybe there'll be some more questions for both of you, but I'd like to introduce Nancy Leland, technologist. Hi, Nancy. Thanks for coming. We're excited to hear your overview.
Hi, everybody. So do I have share screen
Yeah.
capabilities? Okay. So I have a PowerPoint for you. Here we go. Can you guys see that?
Yes, you can.
Yep.
Can you put it in screen show, slideshow mode? Or do we have to do that?
We're getting there. Let's see. Slideshow. There it is. Here we go. You guys seeing that?
Yep.
All right, great. All right, so I'm just gonna give you an overview of what I did, which is kind of a companion to John. As some of you know, he and I have been working together since 2021 on White Pond. And, we've learned a lot, or certainly I have, and I think John has too, about just the cyanobacterial populations in White Pond, which was my objective, especially for 2022, was just to understand what was going on with the dynamics of it. So anyways, let's go through that. So when we started the project in 2022, which is when I got more involved, we have this beautiful recreational site for swimming and boating that has this history of excess cyanobacterial biomass that has resulted in these the beach closures and the public health warnings, which is not what the town wanted, or the people that were living adjacent and using it as, they're not regulated swimming, but they obviously, want to go swimming off their docks. So that's where we came in, trying to do something about that. All right. So I don't know how familiar you folks are with cyanobacteria in general, so I'm just going to give you a little bit of an overview of that so that you can familiarize yourself with what it is that I was looking at in particular. So there's a lot of sort of common cyanobacteria that we see throughout New England that we typically get used to having to deal with in our lab or otherwise as we're doing our monitoring, some of which we know is in White Pond. And I have those outlined here in the red boxes. So based upon what I was doing quite a few years ago with Carmen, we knew there was Dolichospermum, we knew there was Microcystis, and we also knew there was Warnicinia. And so these are very common, wasn't surprised to see them. Included them as we went along, trying to sort of cobble together what was the story of White Pond back about, well, it was probably 5 years ago or so we actually started looking at this. So I had a little more history about what was happening with the cyanobacteria in White Pond and what was actually there. And when we start thinking about cyanobacteria when we're in the lab, one of the first things we look at is what is it that we're dealing with in terms of the ecology of the system and these different organisms that we're dealing with. So we have to think about their morphology, how big they are. So we deal with what I call, size distribution, particle size distribution. So they can range in size anywhere from a couple of microns in size up to thousands of microns in size. But one of the nice things about that is that you can almost put these guys into groups based upon their sizes. And we do that. We have very small ones, we call them the picocyanobacteria, and then we have very big ones that have gas vacuoles, so they tend to form these scums, and we like to call them the bloom forming cyanobacteria. And there's definite reasons why we like to break them into these different groups, and a lot of it is because about the toxins that they produce, believe it or not. And so for cyanotoxins in White Pond, what we were doing this year is we were looking at two in particular that happen to be the bloom-forming cyanobacteria, which is the Dolichospermum and Microcystis, and I'll get into that in a few minutes. But those are the two main types of cyanobacteria that we're looking at. But what's more important is about what we call the ecotoxicology of these particular cyanobacteria. And the reason being is that they have preferences for the toxins that they produce, and they tend to be kind of associated with it. So if somebody tells me I have microcystis in there, and they know they've gone through the process of looking under a microscope, and they'll tell me, oh, we have a microcystis problem, then my immediate first thought is, well, then you're dealing with microcystins, and you'll see that on the far right-hand side over here. If they tell me that they're dealing with Dolichospermum, which is also one of the cyanobacteria that is in White Pond, then at this point I'm thinking about something more associated with one of the other toxins. Anotoxin A has been associated with Dolichospermum, and one of the reasons for anotoxin A, actually the name, and Dolichospermum used to be classified as Anabina. And so when they came around to naming the toxin, they kind of named it close to what the genus was that was producing it, and it was the Anabina was producing this toxin, so they called it anotoxin. But now they've changed the name. I suppose if you went back, you might call it something else, like, I don't know, something toxin that begins with a D. But anyways, that's the history of that. But so those are the two main toxins that I personally would be thinking of as I'm dealing with the populations in White Pond. And that's pretty much what I focused on as I was sort of working my way through the season, was what was happening with the Dolichospermum population, what was happening with the Microcystis population, what might we be having in terms of a toxin profile. Is it anotoxin or is it microcystins? And that's how we typically approach all of our projects, is putting things into these very broad general categories about the different toxins that we have. The one thing I do want to say is that these toxins are very unique as well. They're just as unique as the cyanobacteria that produce them. One of the things they have in common is they're all water soluble, so that's kind of nice. They do have different modes of action in terms of what they affect. More importantly, the thing that we think about is how fast you can see an effect from these toxins. The anotoxin A is known to be an acute neurotoxin. So if you come into contact with this anotoxin, not necessarily by choice, but by happenstance, and you have a very high dose of it, which is, we see that mostly in reports with the animals, like the dogs that are drinking the water along the beach and unknowingly are ingesting large quantities of this stuff that happens to be there. The effects are very, very swift, and actually within a matter of 30 to 45 minutes, if they have enough of this and they ingest enough of it, we have reports of them dying. because of the exposure to this toxin. So, in terms of a risk profile, the antitoxin has a much higher risk for shorter exposure time. The microcystins are a little bit longer, they're more of a chronic effect. So I don't tend to worry about acute hepatotoxic exposure. It's possible. You have to get an enormous dosage to have that response. So that doesn't worry me as much. The BMAA that you see over here on the far left, it's a neurotoxin as well, but that's also a chronic effect toxin. There's a lot of research being done on that. And if you read into the literature, you'll see people talking about the correlation between exposure to BMAA and ALS. So that's people get very excited about that. But I'm more concerned about right now, anyways, people unknowingly being exposed to the antitoxin and having problems with that. So one of the things I do want to say is that, one of the things that we need to know, that saying, the more we know, the less we know. Well, that is definitely true for the stuff with cyanobacteria. We've been studying cyanobacteria for a long time. There's been a lot of work done on it. But, we still don't really understand all the human health and ecological risks associated with them, especially with this picocyanobacteria group over here. These are the really small guys. That's one of the areas of a lot of active research right now. After many years of studying not just antitoxin, but all the toxins, we still do not really have a handle on what the final trigger is for toxin production itself. So you can have something that is what we call being toxigenic, which means it has carries the genes capable of producing the toxin, but we still don't know what that switch is that triggers the production of toxin itself, the cyanotoxin itself. So, believe it or not, we're still trying to figure that out. And I started my master's in 1996, and you'd be surprised. Some of the things that we were dabbling around in 1996, people are coming back with now like it's something new. And I'm like, wait a minute, we were dealing with that in 1996. We've had the same questions. Are people still wondering about this? So yeah, we still don't know, which is a little scary, but there you go. A lot of these toxins, there's a lot of theories out there about why they're even produced. It's one of the things that we're hoping that if we understand why they're being produced, we could understand more about when they're being produced. And that could help us manage our resources better, is if we knew why they're being produced. So, there's this whole chemical defense theory that's going on, that these toxins are produced so that the cyanobacteria can get into what we call chemical warfare between each other, that it's there so that one can take advantage over the other and create an ecological niche that allows it to dominate for a while, but eventually the other one produces its toxin and then it takes over. So, who knows? That's very possible. So that's the whole algal defense theory. they're still working on the concept of this being a micronutrient scavenger type of a molecule that is actively excreted outside of the cell to go out into the water column and harvest micronutrients and bring them back in to facilitate the process of growth. And that's mostly associated with iron being the micronutrient target that these cyanotoxins are looking for to go out and get. So that was a pretty popular theory back when I started my master's, and that was 30 years ago. And apparently there was just this wonderful PhD thesis that was just released all about iron scavenging. So, you know, hey, there we are. We're back 30 years and we're still asking the question about iron. So, there it is. And then there's also the whole idea of these toxins being a source of excess of nitrogen. They're nitrogenous based molecules. Nitrogen is a good source of energy and nutrients. So it could be some kind of a stockpile when they get nutrient deficient, and they use the what we call as toxins, but they look at it as some type of a resource to use when it's necessary throughout the process of whatever their growth or changing environmental conditions or establishing a niche. So all these things are still up in the air. So obviously there's a lot we don't know. But you know, we look at it like, all right, we don't know all these things. We're good with that. Let's just watch and see what they're doing, because we have a concern because they produce the toxins and we don't want to come in contact with them. So let the scientists figure out all this other stuff. But I just thought I'd share that so that you guys understand that if you think that we have all the answers to everything, we certainly don't. But we're having a lot of fun trying to figure that out, keeping us all very busy. Okay, so questions that people tend to come to us with in terms of, you know, if they have a problem with cyanobacteria and they're wondering what's going on, typically the questions are, you know, am I going to have a bloom? And we call that excess biomass, not necessarily bloom conditions, because a lot of people are chasing things that appear at the surface and become a surface accumulation. We like to think of that as excess biomass because there's risk associated when you don't necessarily see everything floating at the surface. It can be a lot going on in the water column, and that goes back to what John was finding with his apod. Is he was working on, you know, all of the movement in the water column and what was happening with that. So all this stuff is below the surface. It's not at the surface creating this bloom, but it's definitely biomass that's there, that's capable of floating at the surface, but not always. So that's what you want to be focusing on. And then what are the toxin concentrations that you might expect to see associated with this stuff? Because like I said, when we were looking at the cyanotoxins and are they acute or chronic, that's really important to know. And then for some folks, how might the toxins be affecting the ecosystem? Not everyone is interested in an ecosystem approach to working with the cyanobacteria, but that was what was expressed from the town, that they wanted to take an ecosystem approach, which I'm really grateful for. So we can do that. And so what we've done is we've developed the cyanocasting program, and it looks at many different facets of what we can do for metrics and the types of questions that we can ask of ourselves and potentially get answers for based upon a monitoring program that is has to follow these golden rules. We have developed this over, quite a few years, but whatever we do, this monitoring, it has to be easy to use and low cost, reliable, repeatable, and transferable. So it's no small charge for us to say, let's come up with something that we think can work. And the reason being is when we're working with people who want to do a cyano monitoring program and have these issues, we want to teach you guys how to fish. We don't necessarily want to be back every year doing all your monitoring for you. We want to teach you how to do it yourself so that eventually you can take it on yourself. And if you need help with something, we're always there to do that. But we prefer to have you sort of internalize it, make it part of your regular monitoring program that you can then go on and use and interpret yourself and make your own decisions about what you're doing with managing your resource. So that's the challenge. So we have some pretty simple methods. We've been testing them now for, it's going on, believe it or not, something like 7 years now that we've had this in the field, working with citizen monitors. But one of the first things we do is we use light microscope, and we can look at what's going on with the diversity index within the cyanobacteria itself. So we're looking at the composition and dominance of what this bloom-forming cyanobacteria is at the genus level. That can tell us a lot right away. Like I said in the introduction, my first question is always to people, well, tell me what it is. And what that means is get out your microscope and take a look at it. And once I do that, and people put it into a category for me, we can start having our conversation. So composition and dominance is always first. And then we have a series of samples that we like to use. These are the 3 samples that you see here, what we call the BFC, the whole lake water in less than 50 microns. These are the 3 samples that are fundamental to the program. And this is the information that we need, like at a minimum, to be able to start having a conversation about what's going on with these different populations. You can always add to it and make it more complicated, little more refined, more information, but it takes more time and it costs more money. And sometimes you wonder if there's a value-added proposition to this whole thing of committing more resources to what you're doing. So that's why we ended up with working with these 3 samples, because we find that we get the most bang for the buck working with these 3. And we can do a lot of interpretation and tell you a lot about what's going on just working with these 3. And then we also do the fluorometry, and basically what that's looking at is we are doing measurements, fluorometric measurements of the sample itself, and giving us a measure of the cyanobacterial biomass. And that has a great advantage, because if you look at almost any program, you'll see that they're asking you to do cell counts. And cell counts are There's a lot of variability associated with it. It can be very expensive. Requires you to send it to an outside lab. Typically, the fluorometry you guys can do yourselves. And it requires a relatively well-trained personnel to be able to do that properly in terms of doing the cell counts themselves. So we like using this pigment. It's an accessory pigment. And it measures biomass as well. So we were like, why wouldn't we be using fluorometry rather than these cell counts to tell us about biomass? Because that's ultimately what we need, is knowing how much is there. That's what biomass means. So we have the ecological indicators. These are our metrics. This is where we can get to. We can look at how much biomass there is and look at how fast it's growing. It's no different than, the farmer that's sowing his seeds, and he goes out and he says, well, I know I planted this many seeds and I made these many rows, and how much is actually coming up, how much sprouted. What he wants to know is what's his standing crop going to be. Well, we want to know how much standing crop and biomass there is too. These are plants. They're no different than, well, they're actually bacteria. But we want to know how much is there too. So we can look at the growth rate and how much biomass is there. We can start to figure out how much toxin might be there associated with that. And then we also want to know, sometimes people want to know what's going on with the fish, and other things that are going on within these ponds. So we can tell something, believe it or not, we can tell something about what's going on with the cyanobacteria based upon what's happening with the fish population, because they can be a very big influence on the whole structure of the whole ecology of the system, based upon what the fish are up to. So that's always very interesting to do a trophic influence project. It becomes it's a lot of fun. It's a great puzzle. If you like to put puzzles together, you do a trophic analysis. If you don't like to do puzzles, don't bother. That's all I can say on that. So the previous monitoring that we did, John was mentioning it back in 2021. I was working with him and using the cyanocasting program. And John had asked me to come in and use our monitoring program that we have to assess the effectiveness of the APOD system. That was back in the fall of 2021. And at that time, we identified Microcystis species as the dominant cyanobacteria at that time. And that was really helpful for me to understand what I might be getting into in 2022. So it was really worthwhile that I was able to get out there. So one of the things I want to show you here is what's called the bloom-forming cyanobacteria, the BFCs. So in his trap here, from his apod, you can see that it's all floating at the surface. Well, those are your bloom-forming cyanobacteria. They float, and he's harvesting them from the surface. So those are the BFCs. You'll see a lot of my data. This is how I organize it when I'm doing an analysis of my samples, is we sort of create these bar graphs of our data, of our samples that we're using, and we look at comparison between our whole lake water, which is, this clear area out here, that's my whole lake water, and what's going on with my BFCs. These are the guys that float. And so you'll see this in my graphs that this is the blue bars. And I'll be talking about that more, but that's really important to see that there's a lot more information coming out of what you see here for these blue bars that represent my bloom forming cyanobacteria. A lot more than what you see with perhaps the whole lake water sample. And that's part of what we do for our analysis is we're using this sample to tell us something that we wouldn't normally tend to see if we just use a whole lake water sample. And we find that it gives us a lot of value added in our interpretation. And it tells us a lot more than we would otherwise. So our White Pond 2022 project, the main objectives from the town, they really wanted a proactive monitoring program. They wanted something that was going to hopefully tell them in advance what was happening at the beach in particular, but in White Pond in general, rather than being a reactive program, which was waiting until they had a problem, waiting until they had to say that it was necessary for beach closure, just being a little bit more on top of things so that they could make decisions ahead of time. And that's what we really wanted. We also wanted to understand what was the risk that you might expect to the cyanotoxin exposure, and how are we going to do that? So we have some metrics that we've developed. We've basically developed some really basic algorithms that we use to tell us about what's happening with the increase in the risk. And that's what we call the growth rates. We can do growth rates and estimate toxins. So the way that the 2022 project was structured, we were looking at 2 tiers. We wanted to do tier 1, which was looking at cyanobacteria and recreation and how those two were relating to each other. And then there were some questions about trophic influences and what was happening with the fish. So we were doing both of those. So I'm just going to give you a quick summary of the main 2022 observations. We started out with 5 different sampling sites. And you'll see that on this graph here, White Pond, where we have these 5 red stars. Satcham's Cove, we have a deep site, we have the town beach, deep site 1, deep site 2, and Thoreau's Cove over here. And you'll see all that data over here on the right-hand side. What you might notice first is that we actually started in May. And so we were out there for a couple of weeks and basically nothing was happening. That's why there's no bars there with any data, because they were all coming in so low that you would basically be calling it a non-detect. And what we started seeing happening at the deep sites and at the cove sites we were going out was that we were starting to see the BFCs. Okay, my favorite friends, the BFCs in that BFC sample. It started to sort of come alive and basically was like, okay, the BFCs are here. They are in the water column. They're coming up. They basically live in the sediment over the winter. That's what they do. And so once it gets warm enough and there's enough light, you'll basically see them starting to rise to the surface, because that's what they do. They have gas vacuoles, and they will create the gases had come to the surface. So what we were seeing basically before we even started monitoring at the town beach was that at throughout the deep site and the coves was that these cyanobacteria were coming out of the sediment. They were done with their overwintering stage, and they were starting to basically occupy the water column. And the coolest thing was, for me anyways, was that I saw a lot of spatial homogeneity there, between all of these very different sites. And what that told me was that it's not just isolated to one particular location, which was one of the first questions. It was like, no, it's the whole water column is becoming, saturated with the cyanobacteria, which is what John was talking about, that there's a lot of influence from the wind and the water movement itself because of the hydrodynamics of the system and the morphology of the pond that are creating these spaces for the cyanobacteria to accumulate on the surface. But otherwise, they're in the water column, and they're pretty evenly distributed throughout the water column for almost all of the summer. So it was a very interesting sort of way to think about, what is going on in there, and that this is just what's going to be happening with the cyanobacteria and white pond, and this is what its profile is like as far as that's concerned. The timeline was kind of interesting. June 1st, when we first started doing the sampling, is when we saw the dolichospermum came up out of the water off the sediment first. That was June 1st. So I'm going to be really interested to see if it tends to, sort of rise from the sediment sooner than something like microcystis. I've never done a study like this where I'm looking at both of them at the same time to see what is coming up first. But the fact that dolichospermum was the first one to come up sort of, it's one of those things I could do some research on if I had some extra time. But kind of cool that it was dolichospermum first. June 14th, the beach was closed because we had this accumulation, believe it or not, of the cyanobacteria. But June 28th is when we finally were able to document that we had Microcystis aeruginosa had come off the bottom of the lake, was now occupying the water column and starting to do its thing as far as moving around and showing up at these different sites and being in high concentrations by the end of June. So that was very informative of when you might expect to see these dynamics happening. You'll see that that's a consistent thing that was happening at all of the different sites. If you look at these graphs, again, the blue is the BFCs. I want to bring your attention to the fact that you really can't even see on here what I would normally be showing is my whole lake water sample. John mentioned that the water quality is very good in White Pond, and that was confirmed by me as well. Basically, in terms of using the equipment that we use to keep track of these guys and be able to quantify their biomass, we are actually below the level, what we call the level of detection for that piece of equipment, to be able to even say that there was cyanobacteria in the whole lake water. So what that means is if you were getting a sample and you went out and you said, well, let me just get a whole lake water sample, see what's going on, you'd say there's nothing going on. Gee, it's really clear. The Secchi desk looks really good. The whole lake water is, wow, that number's really low. But when you look at the VFCs, you'll see how very active they are. They're coming up and they're actually accumulating along the beach, and they did. By the end of June, we were able to sort of narrow our work down to 3 sites. We didn't need to do the 5 sites anymore, which was nice because now we could focus in on 3. The town beach, Thoreau's Cove, and what we call Deep Site 2. So if you look at John's sort of his drogue information, you'll see that where we have these 3 monitoring sites matches up really well with what you might expect based upon what John was seeing for the hydrodynamics of what's going on in White Pond. So we have 3 sites now that we collected data for the whole summer. These are the proposed monitoring sites for next year. And these are the 3 that probably should be monitored at a minimum going forward to give you guys the best possible information to be able to know what the monitoring should be. One of the things that we did find was that for most of the summer, that the Dolichospermum species dominated White Pond, which is a little different than what we saw in 2021. 2021, it was mostly Microcystis. So in terms of the algal defense theory and having chemical warfare between Dolichospermum and Microcystis, for 2022, Dolichospermum won the battle. But these guys tend to be, change your partner, so one year it'll be Dolichospermum, the next year it might be Microcystis. You never quite know what all the conditions are to be able to say which one is going to dominate. But if you watch closely enough, you'll get to sort of, get a good idea of what's going on with the dynamics there. So anyways, 2022, it was Dolichospermum. So one of the other things that we did do was we wanted to take a look at what was going on with the zooplankton population in White Pond, and see if there was any major issues with trophic influences. The trophic structure, basically what it does is it just wreaks havoc with the whole food web. And it just unsettles everything about all of the dynamics of the energy flow through the system that you would normally expect to see with food webs. Kind of locks things into place, creates a bottleneck, and that's what you're seeing here in this diagram. At the top to the right-hand side, the bottleneck that gets created because of the presence of the fish. Rather than letting the energy out of the system as you would normally expect in a healthy food web model. So they kind of create a little havoc going on there, and creates problems of its own. But anyways, so what we can do is we can look and see what's happening with our zooplankton, and they can tell us a lot about what's happening with the fish and whether the system's in balance. And so there is a very healthy zooplankton population in White Pond, at least for what we saw last year. You have majorly your what we call the microcrustaceans, Daphnia species. These guys are very commonly found. They tend to sort of be the drivers, the kings at the top of the zooplankton list. Once you start upsetting what's going on with the Daphnia, things kind of devolve from there, or they certainly can. But what this distribution is telling me, different sizes of them and 3 different types of Daphnia that were there, was that it's a very healthy system in terms of the zooplankton population. So I don't think that there's a really strong trophic influence, and if there is, it's very mild. And there's definitely fish in there. Obviously, there's a native fish, but then it's also stocked as well. So, but what the zooplankton are telling me is that everything's a-okay as far as that's concerned. So that's what's happening with the zooplankton population. So our summer 2022 findings, there's a lot of homogeneity of the cyanobacteria within the water column itself. Like I said, it's highly subject to wind and water movement. And so you'll see these accumulations. This is a picture at the town beach. And you'll see these accumulations of the cyanobacteria being blown into the town beach, creating scum. You can see them right here along the beachfront. I would definitely expect to see the same behavior next year. There's nothing that would tell me why we wouldn't continue to see the cyanobacteria there. I know John is working very hard trying to remove them, and that's a good thing. But until such time as we're able to harvest a lot of the cyanobacterial biomass out of White Pond and have it disposed of safely, that they're going to continue to be there. Part of the reason why is that they can live in the sediments for a very long period of time, and they just need the right environmental conditions, and off they go, and off they grow, and off they come into the water column. So so they can be there for quite a while. They've actually done sediment core studies where they've gone and done deep cores. It's called paleolimnology. Deep cores, the cyanobacteria will be, somewhat dried out, but you put them under the right conditions, a little bit of nutrients, a little bit of light, and they come right back to life, and off they go. So my guess is there's quite an interesting profile to be had in your sediment cores. If you looked at that, you would find them there. So there's nothing to sort of hold them back from continuing to go through their life cycle every year, until such time as they're either used up or they get removed, or some other sort of treatment to quote unquote stop them from growing. Anyhow, so I definitely want to thank the town of Concord for funding the work that we did. It was really very interesting. It was challenging, confirming as well of a lot of things we've already been doing, but has its very own interesting story to why we're seeing these recurring blooms and accumulations in this beautiful system that is otherwise classified as an oligotrophic system. So anyhow, so thanks very much, and I'll be happy to answer any questions that you have.
Thank you, Nancy. So, anybody want to ask a second question? I have none. I have a couple of questions, Nancy, up there. I saw in your report that you discussed the zooplankton themselves at different times. Was there any ability, or did you have any method to measure them? I mean, like how dense they are? Because remember in 2021, we took samples and we couldn't even find them sometimes under the scope. So I was wondering if you can actually quantify the presence, or if you had.
Yeah, so what we decided to do this year is that would have been a biomass study for the zooplankton. You can very easily do that. So what we had originally thought we might do a biomass estimate of the zooplankton, but then what we decided to do was to basically do a catalog and start building basically, you know, a database of the zooplankton, and then taking that and trying to get create a sort of like a little training program, or you know, outdoor experience to work with students at the high school, so that, or the junior high or elementary or whatever, but use it as a way to get the kids down on the water to learn about aquatic ecology, and you know, collect their own zooplankton, or have someone bring the zooplankton into the classroom, and do your own IDs and get your own idea of population structure. And then if there's a need to do the zooplankton biomass, to put the time and energy into it, because you would only really want to do a biomass estimate, quite honestly, if you're looking to see the overall impact of a trophic cascade. And there's no, to me, it wasn't worth it. It was like, oh, I would much rather take the time to put together a program for kids and get them all excited about even getting down to the pond and going and collecting samples, bringing them into the school, doing their IDs, and, you know, just working up like a whole aquatic ecology curriculum around that.
Okay, so thank you. And the reason I was asking about that is because, as you know, this year we did not have the rainbow trout stock in either the spring or the fall. And some of the papers we read and we've seen, you know, discuss this possible connection. So now if they stop in March, April, which it looks like they're going to put rainbow in this time, based on letters we had previous season Fish and Game. I wonder.
Well, yeah, it's kind of hard to say, you know, that was just 1 year of, you know, not having it stocked versus getting this initial data on what was even there for the zooplankton. So one of the things that I noticed about this was in the sampling for the zooplankton, when you have the zooplankton and then you have the fish, and if you have other types of fish other than the what's stocked, in an oligotrophic system, what happens is the zooplankton will sense the presence of the fish, and during the daytime, and they go hide. they go for a visual refuge, and that's what they basically did. And they'll typically seek deeper waters, deeper than where we normally sample for. And the reason why we know that is that we went out one night because we were actually trying to catch a particular type of zooplankton, and you only get them in the water column at night. And we had this enormous population of the zooplankton, and I was like, oh, well, we shouldn't even be doing daytime zooplankton, sampling. This should be done at night. So, if we really want to know what's going on with the distribution of the zooplankton, that's when it should be done. But the fact that we got what we got in terms of the health of that one nighttime sampling was amazing. And I was so glad that we had it because it was incredibly diverse. There was more than what I just shared with you, but it was very, very diverse. And I was like, oh, well, I mean, it might be associated with the stalking program. It might not. That's a total unknown. So I think the best thing to do would be just to start to quantify what's there, just to document it through the summer would be the first step, and to do the nighttime sampling and say, what are we seeing over the course of the season? How is that changing? And that's easily done. You don't have to put a lot of money into it. It's very, very simple. And so that can be done before you even get to the place of doing something with biomass. And then you can have a conversation about it and say, well, this is what we have. Here's our diversity, right? You can do a diversity index and have a great conversation about that, or using that data. So, that's more than adequate to start the conversation. Yep.
Past consultants have said that nutrients and septic system might be the cause in cyanobacteria. And I'm wondering if you feel that way. You haven't really mentioned that and sort of indicate that it's going to happen anyway, even in this later problem.
I'm not completely convinced that as far as the phosphorus loading, continued phosphorus loading that's going on there is which one is contributing the most at this point. I think there needs to be more work done on just internal nutrient recycling. And that's what John was talking about. Basically what happens is that cyanobacteria that you see, the big colonies, once they die, and they do, what happens is they basically are releasing bioavailable phosphorus as well. So depending on how much biomass is actually there, for those guys, and if you can kind of picture it, it's bioavailable phosphorus raining down through the water column before it settles on the bottom, right? And John can talk to you more about that. But it's bioavailable, it's in the water column, and it's part of people. They don't tend to always be considering that when they're looking at a nutrient budget. And sometimes it can be a very large contributor to what's going on with the overall nutrient budget. So if I was going to approach the problem, I would want to know more about what's happening with that part of the whole equation, before I said that it's external.
Another possible cause people have cited is global warming, longer stratification period. What's your view on that?
So what do you think might happen because of global warming? Is that the question?
Well, if the cyanobacteria have been appearing, say, in the last 10, 20 years in this pond, and that's a period when warming has also occurred, or would that be the cause?
Well, they're thermophiles, so they like warmer water temperatures. So it's one of their little ecological advantages that they have for being able to survive and proliferate. So the more we have global warming, the more opportunities there's going to be for them to take advantage of that situation and be able to outcompete.
So Nancy and Don, I mean, one of the targets for our task force is actually, to make recommendations to the town. And, kind of like John's question, if we have any causes, we know some causes, right? Fertilizer, we know the availability of everything that's in the pond already. I mean, what would we, if we're looking at a big program or a program at all, what should we be looking at to start removing things that we have control of? Climate change we can't change. For example, what should we do? Should we add more testing or for this nutrient loading equation? I mean, how do you get that done and understand it better before we say, hey, let's everybody put a septic, take a septic system, not put a sewer in?
No, Beth, that's a really good question, and I think that's something that warrants a lot more conversation. And I know that John had some ideas about that in terms of some of the things that he's more familiar with in terms of the biogeochemical cycling and the sulfur and all of that. I mean, if it was me sitting there, I would want to take the obviously the no chemical approach to trying to look at what to do, and sometimes there's there seems to be like a model that people are using that not that it's a bad model, but sometimes it forgets to look at some of these alternatives. And I think that's where something like John's technology is great, because it's kind of coming in and asking people to take a step back a little bit from everything and not necessarily rush ahead with, a lot of watershed work. I know you guys have done a tremendous amount of work with trying to deal with septic and the runoff and all of that. And all of that is all part of the puzzle. It's all part of the solution. Everything adds to the problem, that you have. But I think they all have to be looked at equally and evaluated to be able to say, yeah, this is, this one is, X percentage, because that's what we'd like to do. We like to make pie charts.
Don, do you have anything to add to that?
Yeah, it's kind of a nutrient budget, and they did this. One of the advantages we have at White Pond is your sister pond, Walden, and academia loves to study both these ponds, and particularly Walden. And there's been an extensive amount of data collected at Walden that speaks to these natural sources of nutrient, including sulfur, actually, and based upon that study, and I think by analogy, by landscape, geology, depth of the pond, there's a lot of similarities between Walden Pond and White Pond. I mean, obviously there's a landfill near Walden Pond. There's some differences out there, but they categorized, where the nutrients are coming from. And they did some pretty extensive testing, and, the dry deposition, like pollen, it was 50% of their nutrient load for phosphorus. They didn't focus much on sulfur. They did test for it, but it just really isn't a metric that most people kind of think too much about. But by looking at their data, they had pretty significantly elevated levels of sulfur in the groundwater around Walden Pond. And they just didn't really, it doesn't really haven't taken any weight yet, I think, with the people out there. But so for White Pond, what you can really do is don't do the prescriptive, assumptive, don't assume, just take some samples and figure out, what is the dry deposition. I think Walden's close enough, you can probably use that by analogy and convert it to like difference in surface water area for how much might be deposited in White Pond versus Walden. But the tree cover, the vegetation is very similar between both ponds. And you can categorize it and say, what are the sources of our nutrients? So at Walden Pond, they didn't describe much of the nutrient contribution to be from groundwater, which is they historically had septic systems there. I remember that mobile home park. I actually grew up in Concord, so I learned how to swim at Walden Pond, and we went to White Pond a lot. So I'm very familiar with it. And so that's what you need to do, Beth, is you just need to develop a table of where are all the nutrients coming from. Don't go by assumption, just collect data. And it's not very expensive to do. Just that most people just don't bother to take the time to do it.
Thank you. Thank you. As I said, that's one of the things we're going to have to come up with, our recommendations. So good advice. I don't see any other questions from the Zoom audience or anything here. So I would just like to say thank you both very much for your very informative discussion and your reports. We really appreciate all you've done. Thank you.
Thank you. I appreciate it too.
You're welcome.
Because we just have to do some other task force business. Or you can stay on too. Stay hang out. Thanks
All
again.
right. Good night.
Thank you.
Thanks, folks. Bye-bye.
Welcome.
I see some eyes that are closing. I think what we can just kind of end, but one thing I would suggest is we have, and Shannon, thanks for hanging out. We had before the cadence of every other week, and then we do this one a month. Do you want to do it twice a month or go to once a month? That moves our and essential too. Thoughts? No.
Yes. Yeah, I think I agree early on. Question on the whole thing.
That sounds, it's me,
Yeah. Some days when we really. Yeah. Okay, so then the next meeting will be March 15th. And then that would be the 11th. I travel so a lot to visit, so that would be one of them, but I'll let you all know. Okay, so March 15th, and then we'll get the next one. We'll just be going for every couple weeks, or at least, I guess, through the year or something. Okay, and then, based on some of this discussion, I hope that was good for all of you, because there are no information to have some to what seems are, sort of, no, I mean, I think that's one of the things that's what we're discussing to you. One of the things we might ask for is that kind of study, right? But I'm not going to want to do Walden and what time, for instance. And I also talked to Mary, we people that are second, third follow along. So I've been on the committee. And how big would it be with it? We don't know. Kind of feeling good. But to use it, well, it would probably be that value. No, I think I think there's data out there that we have to maybe target and get some experts to help. Yeah. I didn't, yeah, no, I mean, that's a question we haven't discussed, with NRC. I know that's the part that we really knew about.
I had a lot of complications with that, supplement of organisms, if it was,
Right.
in many cases, it sounds like you're doing something out more phosphorus, and you're depleting phosphorus in the sediment already just for being weak phosphorus. That might
Because we get a big chunk of it.
It does, but, if you're saying, that's fine, and you see on the website, because you have description of that's fine. It is, and they use that technology with other water. So if there's a 2-inch layer or 4-inch layer, it's stuck in the board and stuff, and I think it would be 1% for the summer. Maybe that is something. Sounds like you recommend.
This would be a lot of phosphorus in the creeks. I think we start off with these. And, I think below the first 4 inches. I don't really see that. Those segments are thick. So you're not going to get all the way down to them.
No, I think this whole point is once we get baseline, right?
so they're not referring sediments. Yeah.
Except for the ones that aren't, right? When he talks about sulfur. These things.
I don't think the sulfur is. So, this sort of thing.
But that's- Yeah, but that's what it's saying is this nutrient loading, this thing that keeps happening, recycling itself. Doesn't really. I think that was his point that sulfur can still be a big problem. And that's what it is. So, that's why I think it's going to study of something would be more valuable.
It's like so that we've had studies in the past, like this. But the charge is, I think, for the consultants, studies that they take, evaluate everything, evaluate, analyze, evaluate, people just thinking about the affect everything. If it was specifically, let's say, contributing area, where, how the future from this, that's for us. All of the things that put on that, right?
Well, I think that's the reason that I come up with it.
Yeah.
What do you
decide that, that's what we still don't know for all these studies. So let's focus instead just on, I don't know what, but, more narrow focus.
Yeah,
It's been in the last study.
I agree. That's what I'm saying. We have the ability to, when we get it, we have the ability to say, one of our recommendations is to do that. One of our recommendations is to do an RFP, this really for another ESS. Whatever it is that we decide, that's how. So I guess that's what I think that it's meaning. Because that's what we have to do. Josh was in the meeting, he's kind of heading it up. And yes, so that's another, that's another thing that we can do. So, if we need money, fundraising or anything, they say, yeah. And we can send money to, and we have a website.
Even our experts that we're talking about, they don't really have, like, a clear, the kind of bacteria come in because of what, they cite, well, environmental conditions are right, so there's no come out. But which environmental conditions, why is it happening now? Why is it happening here and not involved? Is it happening at all? They don't have a stone. They have some existing population. So it's not, you don't have, I don't think a real good knowledge of what's going on.
In 1 year. I have really read the paper when studied right? All for years, people have been studying it.
I mean,
But the work, this work of 1 year, really, John did the follow up. Some people have mentioned depth, clarity.
Yeah, they've done testing.
All the water.
Bill Walker, he's in the service center. But yeah, you're right. There was a citizens group that had been testing for the test. That
But this is
their
the
data
data
was
that we need to create.
I don't think we know the answer yet. I don't think we found, we were hoping we would study the rainwater outfall, work with both of them to get one. But, we so far haven't seen it, right?
the only thing we did see was
We didn't have a
closures
clue,
all year.
but in its experimental design, in the perfect world, we didn't do it because he was there with the APOD, therefore we didn't have a clue. Is that a correlation? We don't know. It was a desired outcome, have improvement, right? You're right, there's so many factors.
Right.
With warm, with, pretty warm, the water was warmer earlier than it has been.
Yeah.
We
We got a couple of new septic systems put in place.
See, there was a bunch of repair, there was
The road.
the road, a lot of work, with drainage, a lot of work, there wasn't a septic system or a home that was having the wash machine run down the river that we fixed, was fixed. So there's a lot of.
No, they usually approve it depending on the perspective. It doesn't mean that it's something 20 years ago, we need to all be one of these. For whatever reasons. But to me, it's different.
Everyone confused over the data, where we're putting all this data, like there's all these different websites, we have a website too, and I know, there were a lot of questions going on, where are we putting all this? The information we want, the information on the facilitation systems, for the program, the data, like, would be on all these different websites,
Well, this data, not only, Board of Health and the Department of Natural Resources are, that's where the budget is coming from for this. So the website has to be up for this.
what, but for all, but to look at it differently, but we want to look at this more than just sales money to data.
Where you get it from?
Where are we going to put it? No, I know that you can get it, but we don't know where to from, I just don't know where to put it.
Which data you have?
The ESS data, the water data, the information on over crossing, information on program. It seems like the EPA guidelines for monitoring, all the different aspects of what we're starting to looking at. It's very.
I mean, Julia has asked that we assess reports under natural resources desk here. Are you asking or suggesting that we have our own area and?
Yeah, I thought we were, that was what we discussed at one time.
Yeah, we're starting for the very beginning. Like, advisory committee stuff that's on there. One, get all that stuff in the one. Even if it's just links to where currently we have. One place.
Yeah. So somebody could take that to town and ask them to repull those and put it in a separate office, but sounds like that's one of the things that is not done.
So I thought Donna had just uploaded a large handful of documents for.
Well, that was what some of the things that he's working on that, but I got confused there, but I thought we were going to have the resource area in White Pond. And if I did the wrong thing, I wanted to stop at this place. I thought we had once for research.
Yeah, do you know if she added it to the watershed page rather than
Well, so
the-
far on the White Pond Task Force, when I looked earlier in the evening, all I had was started. I think he was planning on just sort of jumping on.
Yeah, because I know she did mention, and I don't know if anyone wants to weigh in on this. It's hard for her to, like, she would just kind of be throwing all of the documents on there with no context. And we don't know if that's what your intention would be. But I think it would maybe not be the most user friendly for just all of these documents to be thrown on the page and people not really knowing exactly what it is. So I think it could be useful for maybe some of the documents to be grouped together under a topic, and then we could at least try to break them up by a specific topic on the page.
Yeah, I mean, that makes total sense. I mean, I think that's multitasking, if you're somebody's to work with her to, stakeholders or whatever it's going to be. I mean, I don't know, I think you told me that, some of the stuff to do, like, you're doing this. But yeah, let's just discuss it next, in 2 weeks, and or 2 weeks to work together, to work with that.
Yeah.
Good idea. And then I guess, we, for our agenda next week, we can talk about that. Besides that, right? I don't know what else to say. So there's I'd like to call this meeting, to adjourn this meeting. And, what time is it? Okay, you,
Thank you.
thank you.